series on lipid chemistry, and at the end of this session you should be able to define lipids, classify them with suitable examples, and enumerate the functions of lipids. Lipids are the organic substances which are relatively insoluble in water but soluble in non-polar organic solvents like alcohol, ether, benzene, chloroform, etc. They are actually or potentially related to fatty acids and utilized by the living cells. Lipids are classified in four classes. The first is simple lipids, second compound lipids, third derived lipids, and fourth is miscellaneous lipids. Simple lipids are made up of fatty acid plus alcohol. Compound lipids are made up of fatty acid plus alcohol plus additional group. Derived lipids are those which are obtained from hydrolysis of simple or compound lipids, and miscellaneous lipids are those which do not fall in any category of simple, compound and derived, but they possess the characteristics of lipids. What are simple lipids? They are the esters of fatty acids with various alcohol. The examples of simple lipids are triacylglycerol or triglyceride, which is also called as TAG, and waxes. TAG are the esters of fatty acid with glycerol, and they are uncharged, that's why they are also called as neutral fat. The fat we mostly eat is triacylglycerol, and fat that is deposited in our body as a fuel source, that is also triacylglycerol. Fat in the liquid state is called oil, for example the vegetable oils like groundnut oil, corn oil, etc. And the waxes are esters of fatty acids with high molecular weight monohydric long chain alcohol, and they are not important in human metabolism, but they are widely used in pharmaceuticals and cosmetic industries to make certain lotions and ointments. Compound lipids are made up of fatty acids plus alcohol plus additional group. The examples are phospholipids, glycolipids, lipoproteins. Phospholipids consist of fatty acid plus alcohol plus phosphoric acid and nitrogenous base. And depending on the type of alcohol present in the phospholipid, they are further classified as glycerophospholipid if the alcohol is glycerol, and sphingophospholipid if the alcohol present is sphingosine. Lecithin, cephalin, cardiolipin, phosphatidylserine, phosphatidylinositol, plasmalogen, these are the examples of glycerophospholipid which contain glycerol as alcohol, and sphingomyelin contains sphingosine, and sphingomyelin is the example of sphingophospholipid. Glycolipids consist of fatty acid plus alcohol. The alcohol is sphingosine here, and carbohydrates. That's why they are called as glycolipids. Carbohydrate plus lipids. The examples are cerebrosides and gangliosides. The third example is lipoproteins, which are complex molecules made up of lipid plus protein, and the examples are chylomicrons, very low density lipoprotein, that is VLDL, low density lipoprotein, that is LDL, and high density lipoprotein, that is HDL. The third class of lipid is derived lipids, and these lipids are obtained on hydrolysis of simple and compound lipids. The examples are fatty acids, steroids, which contain steroid hormones like mineralocorticoid, glucocorticoid, estrogen, progesterone, testosterone, etc. Eicosanoids, which are derived from arachidonic acid, which are 20 carbon compound, and the examples are prostaglandins, leukotrienes, lipoxins. Ketone bodies are also derived lipids which are formed from acetyl-CoA. The fourth class of lipid is miscellaneous lipid, and they possess the characteristics of lipids. For example, squalene, which is formed during the synthesis of cholesterol, and carotenoids. Let's enumerate the functions of lipids. They are the storage form of energy. Triacylglycerol are stored in the adipose tissue, and in fasting and starvation, adipose tissue triacylglycerol are broken down to form fatty acids, which are further oxidized for energy. They are structural component of biomembranes. For example, phospholipid and cholesterol, they are present in cell membrane. Metabolic regulators like steroid hormones, eicosanoids like prostaglandins and leukotrienes perform various functions in the body. They act as surfactants, detergents, and emulsifying agents. Also act as electrical insulators in neuron, for example, sphingomyelin. They provide insulation against changes in external temperature due to subcutaneous fat. They give shape and counter to the body. They protect the internal organs due to pads of fat. Help in absorption of fat-soluble vitamins like A, D, E, and K. And one of the important phospholipid, that is phosphatidylinositol, is a second messenger in hormone action. The various diseases which are associated with the lipids and their metabolism are atherosclerosis, diabetes mellitus, obesity, fatty liver, and various hyperlipoproteinemia. In the next video, we will study about fatty acids, their nomenclature, classification, and functions.
Don't skip Nido 3 Plus to help protect your toddler against coughs and colds.
Hello everyone. Today we are going to learn about fatty acids. At the end of this video, you should be able to define fatty acids, classify them with suitable examples, and enumerate the functions of fatty acids. Fatty acids are carboxylic acids with hydrocarbon side chains. They are denoted as RCOOH. R represents hydrocarbon side chain. This is the example of valeric acid. This portion represents hydrocarbon side chain and this is carboxylic acid. Hydrocarbon side chain is non-polar in nature and carboxylic acid is polar in nature. This non-polar and polar nature of this fatty acid make it an amphipathic molecule. In human, monocarboxylic aliphatic acids are found and these fatty acids are usually occur in esterified form. And free fatty acids are transported in circulation bound to albumin. Now let's see how numbering of carbon atoms is done in fatty acids. This is the example of fatty acid. So the numbering of carbon atoms starts from the carboxylic acid. The carbon of carboxylic acid is numbered as 1. Then this is carbon number 2, 3, 4, 5, 6 and 7. And the second number carbon atom is also denoted as alpha. Third number carbon atom is denoted as beta. So this is the C system of numbering fatty acids. What is the omega system of numbering fatty acids? In the omega system, numbering starts from methyl end. So the carbon atom present in the methyl end is denoted as omega 1, omega 2, this is omega 3, omega 4, omega 5 and omega 6. This is how numbering of carbon atoms is done in fatty acids. Now let's see the representation of double bonds of unsaturated fatty acid. This is the linoleic acid, which is unsaturated fatty acid. It has 18 carbons and in the C system, numbering of carbon atoms starts from the carboxy terminus. This linoleic acid is represented as C18 because total number of carbon atoms are 18. 2 means total number of double bonds. So 2 double bonds are present in this fatty acid. Delta 9 represents the first double bond is between 9 and 10 and 12 represents the second double bond is present between 12 and 13. So in the C system it is written as C18 to delta 9 12. Now let's see how it is written in omega system. We know that numbering of omega carbon starts from the methyl end. This is the omega 1 carbon and this is omega 18 carbon. In the omega system, linoleic acid is written as C18 as total number of carbon atoms are 18. two double bonds are there, and omega 6 means the first double bond is between the carbon omega 6 and omega 7. That's why in the omega system it is written as C18 to omega 6. So linoleic acid is example of omega 6 fatty acid. Let's take other example of linolenic acid. This linolenic acid has 18 carbons, and in the C system it is represented as C18. 3 denotes 3 double bonds, and delta 9 shows the first double bond is present between 9 and 10. 12 shows the second double bond is present between 12 and 13, and 15 means third double bond is present between 15 and 16. In the omega system, it is written as C18 3 omega 3. Omega 3 means the first double bond is present between carbon omega 3 and omega 4. So this linolenic acid is omega 3 fatty acid, and this is how it is represented in the omega system. How fatty acids are classified? They are classified on the basis of chain length, total carbon atoms, nature of the chain, and whether they are synthesized in the body or not. So depending on the chain length, they are again classified as short fatty acid if number of carbon atoms are 2 to 6. Medium fatty acids if 8 to 14 carbon atoms are present. Long chain if 14 to 24 carbon atoms are present, and very long if more than 24 carbon atoms are present. We will see the examples in the later slides. Depending on the total carbon atoms, they are classified as odd chain fatty acids and even chain fatty acids. The example of odd chain fatty acids are propionic acid, valeric acid, and even chain fatty acids are palmitic acid, stearic acid, and fatty acid in the biological system usually contain even carbon atoms, typically between carbon 14 and 20.
Oh, and so let's take a look at your activity to analyze
Depending
what
on the
is
nature
called
of
chemical
chain, they are
properties of
classified
your
as saturated, unsaturated, branched, or hydroxy fatty acid. Saturated fatty acid do not contain double bond. Unsaturated fatty acid contains double bond. Branched fatty acid, the example is isovaleric acid, which is the metabolic intermediate. Hydroxy fatty acid, the example is cerebronic acid, which is present in the brain. Depending on the synthesis, they are classified as essential if they are not synthesized in the body and they are taken from the diet, and non-essential are synthesized in the body. Now let's see some examples and sources of saturated fatty acids. Saturated fatty acids are those whose hydrocarbon chains do not contain any double bonds, and they are associated with high levels of total plasma cholesterol, LDL cholesterol, and increased risk of coronary heart disease. The various examples are the saturated fatty acid containing two carbons is acetic acid. It is a small chain fatty acid and the source is vinegar. Butyric acid has 4 carbon atoms and caproic acid has 6 carbon atoms, and they are also called as small chain fatty acid, and the sources are ghee and butter. Capric acid, lauric and myristic acids are medium chain fatty acid, and they contain carbon atoms 10, 12 and 14 respectively, and the source is coconut oil. Palmitic acid has 16 carbon atoms, stearic acid has 18 carbon atoms, and they are long chain fatty acids which are present in the adipocytes. They are most potent in increasing serum cholesterol. And arachidic acid has 20 carbon atoms and it is present in peanut oil. Now let's see the examples and sources of unsaturated fatty acids. There are two types of unsaturated fatty acids. Monounsaturated, that is MUFA, which contains one double bond, and polyunsaturated PUFA, which contains more than one double bond. The examples are palmitoleic, oleic, erucic, and nervonic acid. Palmitoleic and oleic acids are present in adipocytes. Olive oil is a rich source of oleic acid. Mustard oil is a source of erucic acid, and nervonic acid are present in brain. MUFA lower total cholesterol level and LDL cholesterol level, but it maintain or increase HDL cholesterol. Let's see the examples of PUFA, that is polyunsaturated fatty acid, which contains more than one double bond. They are linoleic acid, which has two double bonds and it is a omega-6 fatty acid. Linolenic acid is omega-3 fatty acid, it has three double bonds. And arachidonic acid has four double bonds, it is omega-6 fatty acid. The sources of omega-3 fatty acids are fish oil, canola oil, and flaxseed oil. The sources of omega-6 fatty acids are soybean oil, sunflower oil, and corn oil.
Looking for study help you don't have to second guess? Chegg has millions of trustworthy solutions backed by qualified experts. Go from cramming to understanding with step-by-step explanations designed to help you learn. All the study help you need when you need it is on Chegg. Just search by question and get a response relevant to your course. Chegg has your back with reliable help. Learn with confidence. Learn with Chegg.
New Jollibee Crunchy Chicken Sandwich. Crunchy. Tasty. All about 59 pesos lang.
Now let's see the omega classification and significance of fatty acids. The examples of omega-3 fatty acids are alpha-linolenic acid, timnodonic acid, that is eicosapentaenoic acid, and cervonic acid, that is docosahexaenoic acid, that is DHA. These are PUFA belonging to very long chain fatty acids, and they decrease the level of LDL cholesterol, which is bad cholesterol, decrease the level of triacylglycerol, they decrease the platelet aggregation, and thus they decrease the risk of coronary heart disease. Omega-6 fatty acids are linoleic acid, gamma-linolenic acid, and arachidonic acid. They are polyunsaturated fatty acids. And they decrease the level of LDL, which is bad cholesterol, but they also decrease the level of HDL, which is good cholesterol. The examples of omega-9 fatty acids are oleic acid and elaidic acid. They are monounsaturated fatty acids. Now let's see why PUFA is considered as cardioprotective, that is anti-atherogenic. We know that the lipoprotein HDL, that is high density lipoprotein, is considered as good cholesterol because it transports cholesterol from peripheral tissues to liver, means it prevents the deposition of cholesterol in peripheral tissues. One enzyme, that is LCAT, is responsible for making the discoidal nascent HDL to a spherical mature HDL3. So what is the role of this LCAT? Let's see. This discoidal HDL is made up of phospholipid, mainly lecithin. And this lecithin has arachidonic acid at C2 position. So this arachidonic acid from the lecithin is transferred to the cholesterol to form cholesterol esters, and from this lecithin, lysolecithin is formed. So what is the action of LCAT here? This lecithin cholesterol acyl transferase transfers the arachidonic acid from the lecithin to cholesterol to form cholesterol esters. And by this esterification process, the cholesterol esters move to the core of HDL molecule and it becomes spherical in size, and thus it helps in transport of cholesterol from peripheral tissues to liver by the medium of this HDL, and that's why this arachidonic acid plays a very important role in this process, and that's why PUFA is considered as cardioprotective. Polyunsaturated fatty acid in excess may be harmful. Very high intake of omega-6 fatty acids lower the HDL level, elevate triacylglycerol level, and promote platelet aggregation. Optimal ratio of omega-6 to omega-3 in the diet should be 4:1, but Indian diet has the ratio of 30:1. Sunflower oil has the ratio of 160:1 and coconut oil has the ratio of 3:1. So the coconut oil in this sense is better than sunflower oil. Type of fat is more important than the total amount of fat that is consumed. Which is the best cooking oil? As such, there is no best cooking oil. We should have equal proportion of all fatty acids, that is saturated fatty acids, monounsaturated fatty acids and polyunsaturated fatty acid in the ratio of 1:1:1. The examples that we should consume in our diet: saturated fatty acid, palm oil, coconut oil, ghee; monounsaturated fatty acid, groundnut oil, mustard oil, sesame oil; polyunsaturated fatty acid, sunflower oil, rice bran oil, soya bean, corn oil. So we should have the equal proportion of all these oils in our diet in this proportion of 1:1:1. Let's see the function.
Okay, class? Do you hear me? Class, do you audible na ako? Okay, class. So, you have seen, no, you have taken our quiz 1. So, that gives you an idea of how our biochemistry exam will be. Nahirap ba? How did you find the quiz class? Okay lang? Madali lang ba? Not yet done po. Oo nga. There are how many people who have answered the test? I think there are 21 people who are finished with the test. And the highest score, wait lang, anak. I am, okay. So for the multiple choice, okay, I think most of you got good scores for the multiple choice. Okay. Hello. Okay ka lang?
Ako, okay lang ako, Ma'am Ning. Naunawaan ko po kayo. Kayo naunawaan.
Oo.
Oo, excuse me po.
Teka lang, sorry, sorry.
Ay, okay na. Hindi, Ma'am Ning. Walang problema sa akin. Ikaw pa po ba?
Oh, wala.
Hindi ko yun.
It's not about you, ha?
Ay, oo, Ma'am Ning.
It's a meeting.
Ay, oo naman. Alam ko naman yan, Ma'am.
Mm.
Wait lang.
Class, sandali lang, ha?
Ay, sorry.
Sige lang, okay lang yan, sir. Akin na lang i-ano. Okay, class. So, you're done. There are 26 students who took the test today. And oh, for the... Wait lang ha. And our highest score is... Wait lang. Okay. Students who got high score, Terence Inco, 31. Uh, Wait lang, class ha. Wait. I'll just... So, this is for just for the ano class. The... Hindi ko makuha yung worksheet. This is just for the multiple choice test. I will still be reading the answers to the question. Okay. So, Miss Aguila got 13. Mr. Imbong got 30 also. Miss Mangulabnan got 30 also. Okay. And Mr. Inco got 31. Okay. Now, those who got 29 are the following people. Did you see your scores already, class? After you finish the test, did you see your scores? No.
No po, not visible po.
Not visible. Oo. I still need to check the essay, but this is based on the multiple choice test. So you see how I frame up questions for the multiple choice. So I think you got it well. You understand the question well, and you got the right answer for that one. Okay. So this quiz gives you a good idea of how our midterm examination will be crafted. Okay. So the questions in our midterms exam will be similar to these questions. And for the multiple choice test, most of you got high score. Okay, then, wait lang. Sabihan ko lang kasi hindi ko, nasa na yung aking, okay. 58 minus 25 is 33. So there are 33 multiple choice test items. So, Mr. Buendia got 26. Sean, Mr. Okilada got 29. Miss Lebanan, 28. Miss Toriado, 30. Mr. Besabelia got 22. Miss Del Gallego got 28. Miss Malabag got 28. Miss Viernesa got 22. Miss Peradilla got 29. Mr. Felicilda got 24. And Mr. Saavedra, 29. Miss Fidelino, 28. Miss Gamil, 29. Okay. And Miss Aguila, 30. Miss Algecera, 24. Okay, Miss Cruz, 25. Miss Castellano, 29. Miss Marpa, 21. Miss Baduria, 25. Okay, Miss Fajardo, 29. Miss Kahapin, tama ba? 25. Okay. If I did not mention your name, it means that you got score lower than 20. But it's okay because this is just a quiz, so you have still time to study. Okay, so let make questions. Do you have questions about the quiz? Are the questions easy to understand? I suppose so. Yes, Micaela. Over ilan po yung quiz? Over 58, anak. 25 points yung short answer questions. Okay, yung multiple choice test class. Okay. Hindi ko pa kasi na-check yung short answer questions, which is equivalent to, wait lang, 25 points. Okay. And the first part of the test is equivalent to 33 points. Okay ba? Sige, magtanong na kayo. Ano, mahirap ba yung mga tanong? Wait lang, mali ako. Do you find the questions difficult or not? It's okay. So, 25 over 30, 3. Kung yun ang score mo, that will be 25 over 33. Okay. At 24 points lang pala yung short answer questions class. So, yes, anak. You have something to say? Ano po yung passing ng quiz? So, 60% of 58, anak. 60% of 58, that will be 60 times 15 is 30. 33. Okay, sige. Yes, Miss Pidalino. I-check ko, anak. Pagka sarado na, just send it to my email. Ayun lang ang isasagot ko. Okay? So, ayun. Now, if you want to get high grades, you answer the pre-task and some check-in activities. That will be an additional point for you, in addition to our lab reports. Okay? So, ulitin ko ulit. Last question. Wala naman kayong tanong doon sa quiz natin. Passing grade. Passing grade ay 630 tapos 8 times 33. Undated task po ba sa kan? If you like to, if you want to. If you want to increase your scores. Hindi yun it is not but maybe for those who are frequently absent, it is important that you answer those undated or the pre-task, okay? That will be fine. Nakalimutan ko kasi itong ibigay during the time that you were working for the dental health month, a dental health celebration. This quiz should have been given during that time. But I felt that it is not appropriate because all of you are very busy and you will not be able to focus on the quiz. Okay, so the passing score will be 32. Okay, and if you have a grade of, if you got 29 points for the multiple choice test, you only need how much? Passing score ng quiz po? Opo, anak. 33. I still need to check the short answer questions. Hindi pa included doon yung short answer questions. So the score that I mentioned earlier. Okay? So, okay na tayo? Okay, class. So the last topic is about lipids, and you get a good idea of the different types of lipids based on the, yes, when po yung midterm exam, ma'am? Okay, I want to give the midterm exam on on the 20th. The midterm exam will be on the 20th, class. Okay, on March 20. So uh, ano po yung coverage ng midterm exam natin? Good question. So the coverage of our midterm exam class is are the following. Wait lang. So we have the buffer, pH of common solution, and buffer preparation. So included in this topic is the calculation of the pH of the solution based on the hydronium ion concentration. Okay, the next one is about the buffer system. Okay, how to prepare a buffer, what are the common buffer system in our body. So next will be buffer effects on solution. So as you can see from the quiz, there is a question on the effect of adding hydrochloric acid on buffer solution. What will be the effect of adding 2 drops of hydrochloric acid if the solution is a buffered one, if the solution is an unbuffered one. So that is buffer effects on solution. Now, this is, you need to review our laboratory experiment 1, and then the effect of temperature on buffer solution. Okay, and computation of pH of unbuffered solution and buffered solution. Okay, next will be the cell. Okay, so types of cell, the eukaryotic and the prokaryotic cell. Then we have the parts of the cell. That is module 3. Okay, and the cell membrane. Of course, when we talk of the cell membrane, it includes also the different kind of uh, parts or the composition of the cell membrane. Okay, so after that one, we is our biochemical processes, the processes in biochemical system, transport processes. Now, this includes our laboratory experiment 2. Okay, so this also, yes, anak. Do you have questions? You don't need to take down notes. Why? Because it's the whole module. From module 1 to module 6. I am just repeating the subtopics for each module. So all you need to do is to study our experiment, the results of our experiment, okay? The different reagents that we used, the positive results and the negative results in the different qualitative tests, okay? And then that is very specific for carbohydrates and lipids. Okay, the properties of lipids, also and the classification of lipids. So we still have one experiment on Monday. Then on Wednesday, we will have our midterms examination. Are there any questions, class? Do you have any questions? Hello po. May question po ba tayo? Anak, may question ba? Pangatlo ko na itong tanong ko may tanong. Wala na po. So, okay na yun, anak. So, nagkaintindihan na tayo sa scope ng test. Okay. So, I will check on Friday your answer to the question and then release the scores on Friday also. Okay? So that you can review the quiz. Okay po ba? Okay. Next, meron tanong. May chat po. Ay. Wait lang, class, ha? Nawala ako dun sa isa kong ano, sa isa kong account. Wait lang. Ilang items daw po ang quiz, ma'am. Yung ngayon, ang quiz natin ay 58 items. Okay. 58 items yung quiz natin. Sa reagent, hindi na kailangan yung structures class. Yung mga analyte lang natin, halimbawa glucose, fructose, sucrose. So, tartaric acid, what is the formula for tartaric acid? Teka, baka nawawala na naman ako. Sorry, class, organico pala yun. Lipids pala tayo. What are the samples that we use for lipids? We have olive oil, tama ba? Olive oil, lanolin. Okay. Yes, Micaela, anak. Most probably 60 to 75. Okay ba? Okay lang sa inyo? May short answer questions ulit. Okay? Micaela again. Oo, similar dito sa ating quiz ngayon. Ano? Okay. So, wala na. So, let's continue with our discussion about lipids class. So, you have seen that lipids is not just one class of biomolecules. There are different classes of lipids. We have phospholipids, sphingomyelin, waxes, sterols. Okay, we have simple lipids, compound lipids, derived lipids. Okay, so I'll just post the YouTube that we used a while ago in studying lipids, the different classification of lipids. So let's continue to discuss lipids. Okay, now, so upon getting to know this kind of biomolecules, okay, let us look into the properties of these biomolecules. Okay, so with regards to solubility, are lipids class soluble in water? Based on our experiment last meeting, are lipids soluble in water? Ma'am, no, they aren't soluble in water. Most of them are soluble in petroleum ether. Why is it so? Because our lipids is a non-polar molecule, right? So let us watch the following video to show us the solubility for liquids. Class, sige, let us watch this first. Walang sound. Class, do you hear me? Ay, wait lang. Wait lang. So, do you hear me? So, as I was saying, our alcohol is soluble in, our lipids is soluble in ethyl alcohol, chloroform, and also petroleum, ether, and benzene. But it is not soluble in water. And in our experiment, we have seen that our our lipid is not also soluble in hydrochloric acid. Now, there is a little uh, difference between water and sodium hydroxide. You will see that when you place your oil in sodium hydroxide, it breaks into drop small droplets. Tama ba? Sometimes there is a foam formation. So this indicates that our oil is slightly soluble in sodium hydroxide. Okay, is that clear, class? Class, do you hear me? Class, naririnig nyo ba ako? Ayan, oo.
Yes, bro.
Naririnig nila ako. So, next one is uh, we look into the different properties of our fats and oil. So, what are the different properties of our fats and oil? We look into the color, the odor, di ba? And we try to classify the different uh, uh, oils in our laboratory activity last meeting. So, butter, we don't have butter, but you have seen butter. So, in butter, we have butyric acid. Okay? So, based on the video, what is butyric acid, class? What is butyric acid? So, how many chain of carbon is present in our how many chain? How many carbon do we have in our butyric acid? In the alkyl group, we have how many? We have based on the video that you watch, class, do you still remember? So, sige, baka hindi nyo na-remember. So, I'll just share this tab instead. So, there are three carbons attached to your carboxylate group. Carboxylic acid group. This one is the carboxylic acid group, and there are 1, 2, 3 carbon chain attached to that one. So it is a simple fatty acid. Now, for the coconut oil, structure, how many carbons do we have? Okay. So, this one is our coconut oil. So, you have seen that our coconut oil class is what? Is a saturated fatty acid. Okay. 1, 2, 3, 4, 5, 6, 7, 8, 9. It has 11 carbons attached to our hydroxyl group. Okay? So, It is a saturated fatty acid. Now, compared with our virgin coconut oil, our virgin coconut oil has a double bond and it is an unsaturated fatty acid. And we have what? A test for unsaturation. So, you have seen that our coconut virgin, so this one is a virgin coconut oil, not the virgin olive oil. This is for, but this is a monounsaturated fatty acid. It has only 1 point of unsaturation. Okay. Now for olive oil, our olive oil is a polyunsaturated fatty acid. Okay. There are different so you will be able to see that our olive oil has different unsaturated fatty acid like oleic acid, palmitoleic acid, and alpha linoleic acid. So it is the percentage of olive oil has a higher percentage of unsaturation compared with our coconut oil. Okay, class. I hope this is clear. So, if you see a double bond, a it is an unsaturated fatty acid. It does not have any double bond, therefore it is not saturated, or it is saturated fatty acid. Then what else? What is the chemical structure naman of our lanolin? So let us look into the structure of our lanolin. Okay. So these are our analyte or test sample. So wait lang anak. Mahina yung net ko. So this is our lanolin. You can see here that our lanolin class does not have a double bond. Okay. So it will not give a positive result for the test for unsaturation. So this is the structure of our lanolin, and it is classified under waxes. Okay. So So let us now look into one of the tests for oils and fats. And this is the acrolein test. Sandali lang, class. Sorry, anak. Wait lang. So you have seen the three reactions of fatty acids: hydrolysis, saponification, and the last one is, I forgot, hydrogenation. Okay. Hydrogenation is a re- reduction reaction, okay, wherein the polyunsaturated fatty acid um, receives hydrogen, okay, and becomes saturated. Uh, that is hydrogenation. And as what is mentioned, it is very important for the what you call this one, production of margarine. Okay, now the next one is the saponification. It is a process of making salt. Okay, now one of our product is coconut oil, and many industries are using coconut oil to produce soap, detergent, and the likes. Okay, so uh, coconut oil is a valuable product of the Philippines. Then another one is the first one is the hydrolysis. So it is the breaking. When you say hydrolysis, you break the bonds of the fatty acid, okay, using water. Okay, and you have seen uh, how an ester of a fatty acid breaks down into glycerol and its component fatty acids. Okay, so those are the three reaction of our um, fatty acids. Now, this one, we perform this in the lab, the test for unsaturation of oil. So let us watch this one. Okay.
Ma'am, nakamute ka po.
Sorry, nakamute na naman ako. Wait lang uli. So, doon sa iodine, nag-aano siya. Yung iodine test is good for our olive oil but not for our coconut oil and our waxes. So, another test for unsaturation is the bromine test. We did not do it, but still let us watch this video. Wait lang ha. Seen that bromine reacted with our virgin coconut oil. Okay? So, for the other oil, you have seen that it did not affect, it did not change its color that much. Okay? So, another example, so that is how our bromine test will look like in determination for unsaturation. The faster the bromine color disappears, the higher the degree of unsaturation class. Okay, next one will be the saponification process. Where you have seen how to make your soap. So it's just a mixture between coconut oil and sodium hydroxide. So when coconut oil is mixed with sodium hydroxide, you form a suspension, okay, of salt and of oil salt. And then you filter that one, and that one now is your sodium. That one will be your soap now. Okay, so we'll end our class up to this point. This will be our last topic. So you will be performing the last experiment on Monday. Okay, the acrolein test and the other test. Okay, so I will post our, wait lang anak, stop sharing muna ako. I will post our experiment in our group chat later. Okay, so it's already 4:02. Thank you so much for your time. I just hope that you start reading now. Class, I will be looking into the amount of time that you spend reading in our modules. Okay? So please do read the modules. What you have today is just a sample of some of the questions. Okay? Baka akala nyo yun na yung tanong, hindi pa yun yung tanong, anak. Ha? Okay, so I'll see you on Monday. On-site tayo. Wednesday, on-site also. So, see you class. Bye-bye. Bye.